I'm Daniel Levine, and this is the Bio Report. One reason that cancers can be difficult to treat is that the tumor microenvironment can hamper the ability of drugs to penetrate the tumor and facilitate the development of resistance to medicines. AUM Biosciences is developing targeted therapies that address the genetic drivers of a cancer and can disable defenses that hide tumors from the immune system. The company's lead experimental therapy is being tested in combination with immunotherapies to treat colorectal cancer and other solid tumors. We spoke to Vishal Doshi, chairman and CEO of AUM, about the development of resistance in cancers, the case for combining precision therapies with immunotherapies, and why he believes we can transform cancer from a deadly disease to a chronic condition. Vishal, thanks for joining us.
Thank you, Danny.
We're going to talk about targeted therapies, cancer, and the problem of resistance. I think for listeners, it might be helpful to start with the idea of kinases. What are they? What role do they play in human biology? And what role do they play in cancer?
Yeah, no, thanks for that great question. So effectively, kinases are enzymes that transfer a phosphate group to a protein, while phosphatases remove a phosphate group from protein. Together, these two processes, they modulate various activities of proteins in a cell, often in response to an external stimulus. There are approximately 538 kinases that are encoded in the human genome. And all these kinases, they maintain the cellular function by turning protein function on, while corresponding the phosphatases reverses the action. That's what kinases are. And the recent advances that we've seen in targeted therapies has just given us more understanding and on the fundamental molecular mechanisms underlying the cancer cell signaling. And that has further validated the crucial role of kinases in the carcinogenesis and the metastasis of various types of cancers.
With the approval of Gleevec, the first tyrosine kinase inhibitor, we've seen this move towards targeted therapies and precision medicine in cancer. How effective have we been at changing the way cancer is treated?
So I think there's still a lot to be done. And we have certainly made a lot of progress, as you rightly mentioned about with the approval of Gleevec, and some recent advancements in some targeted therapies. But there's still a lot of work to be done on that. My vision for cancer is really to see if we can convert cancer into a more manageable disease, very much like a diabetes or a infectious disease, where you don't accept a 20% response rate, do you, for a fever? You need the fever to go 100%, not 20%. So I think there's still a lot to be done, but great advancements have been made. And there are certain problems that we need to address to overcome the challenge that cancer brings in. One additional point I would like to add, Danny, on this is cancer is not a single disease, right? If you break cancer down into subtherapeutic, if you break cancer down into various modules per se, you can actually see there's so many different types of cancers within the cancers, right? So it's more like niche indications that we look at. even within one single indication. Classic example, colorectal cancer has different subtypes, content molecular subtypes, which we'll talk about in a little bit more detail. So I would say great advancements have been made, but still a lot of work that needs to be done on that.
One of the challenges has been the ability of cancers to develop resistance to therapies like tyrosine kinase inhibitors. What happens within the tumor microenvironment that allows it to develop resistance to a given therapy?
Yeah, no, that's a great question, and it comes. What comes to my mind is innate resistance, and innate resistance to various therapeutic interventions is kind of a hallmark of cancer. But at the same time, there is acquired resistance that has become a emerging and daunting challenge to various anti-cancer treatments, which includes chemotherapy, radiation, and targeted therapy. Now, what this does is it kind of abolishes the efficacy of otherwise successful regimens. What happens is the cancer cells, they gain resistance through a variety of mechanisms, both at primary and metastatic sites. This includes the cell intrinsic and the extrinsic factors. But the point that remains overlooked is the extrinsic factor per se. And what happens in the tumor microenvironment plays a very important role in there, and in multiple aspects of cancer progression, and particularly, which is a big problem of therapeutic resistance. And to just put it very simply, the tumor microenvironment decreases the drug penetration. It confers the proliferative and anti-apoptotic advantages to surviving cells. It facilitates the resistance without causing genetic mutations and epigenetic changes. And collectively, it modifies the disease modality and distorts the clinical indices as well. So to overcome all this, tumor microenvironment, it plays a very important role, and an essential strategy to overcome cancer resistance is to actually target the tumor microenvironment. But more importantly, we should also focus, and we are also focusing on improving the therapeutic outcomes through precise intervention, what you call it as targeted therapy.
One of the approaches we've seen to addressing tumor resistance is using combination therapies. What's the case for a combination approach?
So the way, I mean, if I were to draw a parallel to this, right, when in 1960s and 1970s, when infectious disease was quite a big challenge per se, one drug never did the trick, right? And now you have various infectious diseases that can be solved by having a cocktail therapy per se, or even 2 to 3 drugs all at once. Now, to your point about the case of combination therapy, let's just take an example of drug that is currently out in the market of what we call it as Keytruda, right? Now, Keytruda as a monotherapy versus Keytruda as a combination therapy is actually seen that combination therapies as a whole, it kind of takes a very holistic approach towards cancer treatment. You're not addressing one problem, but you're addressing another problem that might arise from the treatment that you are giving, but you're forward-looking that particular problem and trying to address that to a combination therapy. Hence, at AUM, we are actually looking at combination therapy as the future of cancer treatment, to not have that resistance develop. And if there is any resistance that develops, you kind of address that by developing a combination treatment.
You've talked about the potential to transform cancer from a deadly condition to a chronic disease. Can you explain that?
Yes, this is very close to our philosophy that we follow at AUM Biosciences, and the example I gave you earlier, if you get fever or if you have diabetes, you don't want to be in a situation where the doctor tells you that your only 20% of your fever is going away, right? You want the doctor to tell you, take this medicine and all your fever is going to go away. You're going to get better, up and running quickly. But unfortunately, in the case of cancer, and there is a lot of progress that is made, but unfortunately, in the case of cancer, the quality of life gets impacted significantly. And what we at AUM Biosciences would like to think is if we can convert cancer into a manageable lifestyle disease where people can live with it. It's not the end of the world. It's not a death sentence when somebody gets cancer. But it's really to manage your lifestyle, to manage your diet, to manage your medication, and so much more that goes into it. Therapeutic treatment plays a very important role in it. And if we can actually come up with a combination therapy, or even a monotherapy that enables a patient to live a healthy life after treatment, that would be a game changer in how cancer is perceived out in the open, as a potential death sentence as well, right? So we don't want that to happen, and we really want to work towards patients' support to try and see if we can come up with therapeutic treatments that improves the quality of life for patients as well.
Your lead experimental candidate is a MINC1/2 inhibitor. What is MINC1 and 2, and what role do they play in cancer?
Yeah, so MINC1 and 2 are the enzymes that play a very important role in phosphorylation of EIF4E. EIF4E is overexpressed in majority of the cancers, and phosphorylated EIF4E is overexpressed in majority of the cancers, including solid tumors and liquid tumors. And MINC plays a very important role in that. It's a very crucial pathway from an RNA to protein translation. And if you inhibit MINC1 and 2, we have actually seen that it plays a very crucial role in inhibiting the phosphorylated EI4E, ultimately impacting the RNA to protein translation and ultimately impacting the cancer progression to that effect. And hence, we are developing a small molecule for MNK1 and 2 with a specificity of 99.5%, which has always been a want from a MNK1/2. And at the same time, we have conducted some recent studies that validates the mechanism or the pathway that we are targeting to see whether this is an interesting target or not. And we are very excited about the fact that is moving into a phase 2 study, a global phase 2 study with patients participating in this study in a very imminent manner as well.
Well, what is the lead therapy and how does it work?
So as I explained to you, AUM001 is a specific MNK1/2 inhibitor, and it kind of addresses two major problems, right? So first, as I highlighted earlier, specificity has been a big issue with MNK1s. In our case, we have a 99.5% specificity. The second differentiator and second issue that we are trying to resolve over here, it's converting cold tumors into hot tumors. And in AUM001's case, we actually have seen solid target engagement to validate the hypothesis that I just mentioned about converting cold tumors into hot tumors. And when you give this as a monotherapy, there is some reduction in tumor size, but when you give it as a combination therapy, it has shown some significant tumor growth inhibition in solid tumors that are very hard to treat. And to my point earlier, it's converting cold tumors into hot tumors. And that's what is exciting about AUM-001. The way it works is it inhibits the phosphorylation of EIF40, which is an important mechanism from an RNA to protein translation.
I take it the significance of being able to turn a tumor from cold to hot is that it helps make it visible to the immune system?
That is correct. And no, you're absolutely right. And it also plays an important role in the tumor microenvironment as well. So it kind of addresses a problem of T cell exhaustion. And that's one of the major reasons why a lot of the immunotherapies don't necessarily work all the time, because of the T cell exhaustion as one of the challenges. And what AUM-001 does, it kind of addresses the problem of T cell exhaustion and helps with conversion from cold to hot tumors.
You're looking at this in combination with the immunotherapy Keytruda and separately in combination with the immunotherapy Tecentriq. I take it that's why?
That is precisely the reason why. You are absolutely right. And we are very excited about the combination with Keytruda as well as the combination with Tecentriq. The data we are expecting the data to come in the next few quarters. But yes, you're absolutely right. Is that conversion of cold to hot tumor, and specifically the indications that we have chosen, which I believe we are going to talk about, it plays a very important role in a combination therapy.
Are you looking at 001 as a monotherapy at all?
So we do have some data that gives us some validation around the fact that this drug works as a monotherapy. But as an organization, we are very particular about not setting ourselves up to fail, because we understand that even a monotherapy drug will ultimately have to be combined with something or the other, right? So monotherapy of 40, 50, 60% monotherapy is not exciting enough for us when we are trying to address a big problem of cancer resistance, because then you come back to square one of addressing resistance. So monotherapy to address cancer resistance doesn't look like a very attractive proposition, and hence we are very wedded to the concept of combination therapy. But we do not decline to develop our drugs as a monotherapy. We actually have other products in our other drugs in our portfolio that provide monotherapy data, but that is at the latter stage of treatment, not at early stages of treatment, right? So latter stages of treatment, once the resistance has developed, then it's a different story. But to do it as a first line or second line as a monotherapy only, there's bound to be resistance that gets developed.
You're looking at this in combination with immunotherapies. Are you looking at any combinations that involve other kinase inhibitors?
Yes, interestingly, we do have some data in combination with other kinase inhibitors. But the interest of capital efficiency, in the interest of focus, our immediate focus is going to be with AUM-001 in combination with Keytruda, and we would also like to develop AUM-001 in combination with Tecentriq. And then we have other things on our portfolio that takes a lot of our time. So AUM-601 and AUM-302 also takes up a lot of time as well.
This is a potential target in a broad number of cancers. How are you determining which indications to pursue, and how are you prioritizing those?
Yes, so there's two parts to that question. The first part, how do we identify a lead indication? This is really based on the data that we have generated till now, and where we see as the highest probability of success for this particular drug in combination with Keytruda. We have also discussed this at length with our partners as well. And we've come to a conclusion that we will be moving to develop AUM-001 for colorectal cancer, and that's in combination with Keytruda. The fact that this drug or this particular pathway plays an important role in multiple tumor types, we are also very we take pride in how the clinical trials are designed as well, Danny. We feel very confident on the fact that our track record of conducting over 150 clinical studies allows us to design a trial which is more a basket trial rather than a single trial, single indication, and have different arms to it, where you can decide whether the first readout on the data is good enough for you to advance it to the next stage, or you actually kill that particular arm and transfer the patients or move the patient to a different therapeutic regimen per se. So we depend on the fact of basket trials and umbrella trials on innovative trial designs to help us reach to a proof of concept as quickly as we can and in the lowest amount of capital expenditure as we can.
Known about 001 from studies that have been done to date?
So we have completed our 2 phase 1 studies for AUM-001. One was done in Singapore, one was done in Australia. And some key highlights that we have observed from that: number one, we have seen solid target engagement. At a very low dose, we have seen solid target engagement, which validates the fact that this drug is targeting where it's supposed to target, which is fantastic. We have also seen that at the highest dose in phase 1 settings, we have not seen any form of major serious adverse events. We have seen some minor grade 1, grade 2 serious adverse events, but nothing major that's going to be of concerning. And that was actually very exciting when you think of a combination therapy. You are developing a drug in combination with already a standard of care. You can be rest assured that the SAEs or toxicities associated with the combination therapy, you can be rest assured that not a lot of that toxicity is going to come in from our drug. So that unknown of additional toxicity for a combination therapy is kind of addressed quite significantly. And more importantly, the focus then becomes on the actual patient data that we intend to generate in the coming months as well.
And what's the development path forward?
So we have received a ethics committee approval for AUM-001 from the Australian regulatory bodies. We have initiated a global phase 2 trial with sites in Australia. We will be initiating sites in the US and some part of Europe. We are looking at approximately 110 patient trial. The study has already begun. The first patient in is imminent anytime now, and we'll be happy to share that news with you once that happens. And we expect that the study, which is broken into 2 modules, module 1 and module 2, we expect that the module 1 data readout should come out in the next couple of quarters. That should give us a hint on proof of concept for this particular drug in the coming months. So we're very excited about the fact that we are going full speed ahead on this particular trial and expecting some data coming out of that.
Is based in Singapore. I know Singapore made a big push into biotech and really was highlighting the sector. It's got a rather advanced healthcare system as well. How are you thinking about markets and clinical trials in regard to your geography?
Yeah, I feel that trial cannot be a single country trial, and especially with the thought process at AUM, we have a mandate that any drug that we develop will need to have a US IND right from the outset. And for one simple reason that a phase 1 study done in Asia can be done in countries where there is very solid backing from regulatory bodies such as US FDA, so that the data is actually accepted by the US FDA when we go into a phase 2 study, which is a much larger phase 2 study. And to just close the loop on that, I think we have never been of an opinion that doing a study in one country is a good enough answer for us. And hence, all our studies that you will see are going to be global in nature when it's a phase 2 program that you're looking at, so that you can get a diversity of patient population coming in from the West, coming in from the East, and then combining that to give a solid data pool when we are actually thinking of advancing and getting a proof of concept around it. So our mandate and mantra is really to look at a global approach right from the very beginning, even though we are based in Singapore, but we will be looking at, we are already a global company with presence in Australia, presence in the US, and recent expansion in a small setup in China as well. So we are getting ready for it. Global is the name of the game, not individual country studies.
Did a financing late in 2022 to raise money. Can you explain that transaction and how far will existing cash take you?
Yes, so I am going to share as much as I can with regards to that because we're bound by confidentiality on that. But yes, what's in the public domain, we did a SPAC in October 2022. At the time of closure, the SPAC had $69 million US in the trust. The pre-money equity value on that was $400 million US. We are all aware about the fact that SPACs come in with certain level of redemptions as well. We are actively raising PIPE financing, and we're actually getting some very good interest from the market on our PIPE financing. But what of all these things, what doesn't change is what we are going to focus on even after we become a public company, and that is being a capital efficient company. And to answer your question, with the cash, the existing cash that we have and the various financings that we have done till date, it gives us enough runway for additional 12 to 18 months time. And we will be looking at raising additional capital as we may need to advance our programs further. So very exciting times with two clinical programs, two preclinical programs that we currently have. And then once we generate more data, I'm sure we are going to generate a lot of interest in the market. So do keep a lookout for AUM Biosciences, and we're excited about this.
Vishal Doshi, CEO of AUM Biosciences. Vishal, thanks so much for your time today.
Thank you so much, Danielle. I really appreciate your time.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.